Hello dear student, myself Rominder Kaur and here I am going to start the fourth video lecture of chapter number 7, Evolution. So today's topic we are going to start with Hardy-Weinberg principle. If we generally see in any population there can be different group of organisms, like if we take example of humans. Some may contain dominant genes, few may contain recessive genes, and some may contain the variety of dominant and recessive characters that we call as hybrid species. So if I take here an example of eye color of human beings, some may have blue color eyes, some may have black, or others may have brown color eyes. So these are the effect of genes on the characters of eye color. So if we take in any given population the frequency of occurrence of alleles of a gene or a locus. Now let me define here the locus. Locus means the location of the genes present on the chromosomes. So this frequency mostly remain fixed, means the total number of organisms who are dominant plus recessive plus those who contain both the characters, it always remains same, means it is fixed and the value is equal to 1. So Hardy-Weinberg was the scientist who has proposed a principle based on algebraic equations. He said that evolution will take place only when there are mutations, when gene flow will be there, genetic drift will be there. So certain kind of conditions or factors are responsible for evolution to occur. So on the other side, evolution will never occur if there will be no mutations, no variations in DNA structure, if there is no gene flow, if there is no natural selection, then no certain kind of changes will lead to any kind of evolution. First of all, the definition of allele is the pair of genes. In any population, the frequency of alleles will remain same. If we take example of any character, for dominant we will write it as capital A, capital A. Why not written as one single capital A? Because genes are always found in pairs. If they will not be in pairs, then it will be difficult to segregate them into further generations. Some can be recessive, so recessive will be written as small a, and others which are hybrid, that can be written as capital A, small a. If we take example of pea plant, the study was done by Gregor John Mendel. So he studied pea plant and said that if we took the height of the plant, then we can write down the tallness as capital T, then dwarfness as small t, but when both are crossed with each other, then the resulting F1 generation will always be hybrid, but it will show the tallness character because capital T is the dominant character, then small t. So this principle states that the frequency of alleles will remain same from one generation to another. Either it is first generation, second generation or third generation, frequency will always remain same. Like law of segregation, let's study from this. Here, the color of the flowers is taken as the trait. One flowers which are purple color, when they are crossed with white colored flowers, so purple color is dominant over white, so that's why genes are written in capital P, but in case of white they are mentioned as small p because it is a recessive trait. Now what will be the F1 generation? It will be purple flowers, so it contain 50% capital P and 50% small p, but because capital P genes are dominant, so these color of hybrid flowers, they will be of purple color only. But when these purple colored flowers, they are crossed with each other, this kind of resulting generation will show law of segregation, means 1:2:1, that is a genotypic ratio, and 3:1 is a phenotypic ratio, means 25% will be pure purple colored flowers, 50% will be hybrid purple flowers and 25% will be white flowers. So law of segregation states that whenever the hybrid kind of traits they are crossed with each other, the resulting generation can segregate the number of genes. Hardy-Weinberg equilibrium, because he has taken algebraic equation, he states that when we take the frequency of dominant and the recessive allele, this frequency is always equal to 1. So as in mathematical equations or physics equations you have studied, when we are writing anything is equal to 1 means we want to show that these kind of characters they are showing some constant behavior. So here again, small p and small q are taken as frequency levels, one for the dominant allele and other for the recessive allele. So dominant character is purple and white color is recessive alleles, but the sum of both should be equal to 1. Then there will be no evolution, no such changes will occur that will show something different in them. Means generation to generation there will be two color only, one is purple and other is white. There will be no mixture of purple and white color can be seen from one generation to another unless variations or mutations takes place in the DNA. Now if we take the whole square of p plus q whole square is equal to 1, what will be the equation? It will be p square plus 2pq plus q square is equal to 1. Now how can we relate it with the cross that is shown on left hand side? P square, if we take capital P, so it appears twice. So we have written frequency of homozygous dominant genotype. Homozygous means because both are capital and both are of pure type only. Then we have written Q square. Q square means small p, small p. Earlier we said Q is equal to only the recessive allele frequency, but Q square we are saying frequency of homozygous recessive genotype. Now what is 2pq? 2pq is equal to all the 50% hybrid characters which appeared as hybrid purple flower such as capital P and small p. So what will be the sum of all three frequencies? That will be equal to 1. So you can see here, generally we said p plus q is equal to 1 and the whole square of p plus q is also equal to 1. Then when we open this whole square, it comes p square plus 2pq plus q square is equal to 1. So here all the kind of frequencies they are written. What is p? Frequency of allele A. If you do not want to write capital A here, you can also write capital P here. If you want to show tallness and dwarfness characters of Gregor John Mendel, then you can also write here capital T. These are only for the references, either A is taken, T is taken or P is taken. It depends upon which kind of characters we are showing. But just one thing you have to remember, when you write P, small p, that will be the frequency of dominant allele only. So here you have to write capital A or capital P or capital T or any other trait where you want to show any kind of cross. Small q is equal to frequency of recessive allele. So here you can write small a or small p or small t. As you want, you can take your own choice letters here. Then what will be p square? It is a frequency of homozygous dominant genotype. So obviously it will come twice. P is equal to capital A, P square is equal to capital A, capital A. If I talk about 2pq, so it is frequency of capital A, small a, means a hybrid kind of genotype. Q square is equal to frequency of recessive alleles, so it is small a, small a. So when the sum of allelic frequency is equal to 1, then there is no evolution. But when the sum is not equal to 1, means the ratios they are changing, something new hybrid is being formed, then there are chances of evolution. So let's study what are the different factors which are responsible for evolution to occur. So in this paragraph, they have explained each and everything in any kind of diploid genotypes. Why diploid genotypes? Because we all living organisms are diploid. Those who are undergoing sexual reproduction, we are diploid organisms. We have 23 pair of chromosomes in human beings. So similarly, if we have only 23, that will call as haploid, which are gametes. But when we are talking about any individual, it means there is necessity that we must have diploid kind of genotype. So this paragraph will be clear what I have already told. Now if we see on the right hand side picture, both mother and father, they have capital A, small a. Father is also having capital A and small a. So they have taken capital A is P frequency and small a as Q frequency. What can be the chances of the new baby which can born? That may be P square, means it can be purely dominant characters can be there. Then it can be a mixture of both the characters, capital A, small a, or it can be that both small a can mix with each other and form the recessive genotype. Now, let's talk about the 5 factors which are affecting this equilibrium. So, in this picture, number 1, no selection. If random mating is there, then there will be no evolution. If no mutations takes place, no migration, large population is there, and random mating is there, then it is leading to no evolution situations. What will be the opposite of it? If we select the certain kind of desirable characteristics. You must remember chapter number 9, strategies for enhancement in food production. We have talked about selection of parents, both in the case of plant breeding and animal breeding. So that is a kind of evolution that we are bringing some desirable characteristics in the new hybrid. We are selecting that lactation period in cow must be good, and then we are going to cross it with the bull that should have more vigor and strength. So here we take that if evolution will occur, then which factors are responsible? Number 1, selection. Number 2, mutation. Number 3, migration from one place to another. Third picture, that the species of the frogs, they are differentiated with a canal or a stream of river. So if one kind of species, they migrate to the second kind and they will reproduce with the another kind of species, it will result into new hybrid. So that means evolution will occur. So if population is small, then evolution will take place. There will be no random mating. So you must remember these 5 factors which are responsible for this Hardy-Weinberg equilibrium: gene migration or gene flow, genetic drift, mutation, genetic recombination that we have already studied in chapter number 9, and last is natural selection. So whenever migration will take place from one population to the another, it means this will result into evolution, because genes will change, genes will migrate from one population to another, and new genes will be added to the new population, and old population will lose all the genes which were present in them. So this would be a gene flow if this gene migration happens multiple times. Multiple times means generation after generation. If this is taking place, then it is only by the chance it is leading to genetic drift. Sometimes it happens that the allelic frequency is changed and it is so different from that new sample of population is formed, and it results into formation of new species that we call as speciation. So this kind of effect is called as founder effect. You can see in the picture a change in the gene pool caused by small population migrating to the new area. So this means it has resulted into a drift of the population change. So the new species which will form, they are called as founder. So this effect is called as founder effect. Real experiments have shown that earlier we have studied that when Darwin talked about natural selection, he said that if we keep bacteria in a container under certain conditions of temperature, if we suddenly change the medium or the surrounding of those bacteria, then some of them will survive and other generation will die. This is because few of them, they develop some changes in them and we call them as new species, and they result into new observable characters. So natural selection is a process in which only the better population will survive, those who can enable themselves to produce good progeny. And even the critical analysis shows that the variations which are occurring due to mutation or during gametogenesis, this may lead to genetic drift, which will ultimately change the frequency of the alleles, and P plus Q is not equal to 1 whenever evolution takes place. So natural selection can lead to stabilization, in which more individuals acquire mean character value. So there are three kinds of natural selections. Number one, when the generations become stabilized. So let's this you can see, if I start from the middle one, the number of individuals with phenotype, they are shown on the y-axis, and by natural selection, medium-sized individuals are favored. Neither the small nor the taller sized individuals are favored, only the medium sized are favored. When the peak gets higher and it is narrower, this means number A is showing of the stabilizing and number B is directional, means it is moving to one kind of direction, that new species are produced. In the third one, you can see disruptive curve has been formed. Firstly, one peak is there, then after some time the second peak will form. So this was about Hardy-Weinberg principle, which generally talks about the frequencies of the allele, that around us, if the frequency of the genes will remain same, then there is no evolution. But when the frequencies keep on changing generation after generation, it will result into new species that we call as speciation. Thank you, students.